Is. What is chromatography anyway? Is it some kind of fancy sandwich? But wouldn't that be cool? Chroma sandwich? Anyway, chromatography is a method scientists use to separate a mix of stuff into its individual parts. Kinda like when I separate the good snacks from the weird health snacks in the pantry. Why would you do that? Because sometimes you want to know what's in something, how much of it there is, or to purify a single component. Makes sense, right?
How does it actually work? Picture two buddies at a party. One buddy likes to stick to the couch, that's the stationary phase, and the other buddy likes to move around the room, that's the mobile phase. If you release a bunch of different buddies, some stick more, some move more, and eventually they end up in different places. That separation is chromatography. So what types are there? There are lots, but here's the main menu. Mine is paper chromatography. The old school method using paper is the stationary phase. Cheap, simple, like my first paycheck. Thin layer chromatography. TLC, like paper but on a thin plate coated with silica. Faster and better for quick checks, great for lab folks. Column chromatography. You pour your mix down a column filled with packing material. Stuff comes out in order. Think of it like a long slide for molecules. Gas chromatography, GC. The mobile phase is a gas. Good for volatile, smelly things like when I try a new hot sauce and the kitchen smells for days. High-performance liquid chromatography, HPLC. A pumped, high-pressure version of liquid chromatography. Super precise and fancy. Scientists love it for quantifying stuff. Ion exchange chromatography. Separates based on charge. Do you want positive or negative? Take your pick. Size exclusion gel filtration. Separates by size. Big ones come out first because they can't get into the little pores. Affinity chromatography. Super specific, uses a molecular bait that grabs exactly what you want, like Lois calling me into the kitchen with the promise of beer. What should we think about next? Why do certain molecules prefer the stationary phase while others don't? How could changing the mobile phase totally change the separation? If chromatography can separate things so well, what ethical questions arise when it's used to isolate powerful compounds? Food for thought. Now who wants a beer?